Hi guys, it's Rob from Royal Balls, part 2 of the genetic series, and today we're going to look at cell division and how that plays a role in the way that we produce all our designer morphs. The process itself involves some very complex chromosomes and genes, but the principles that govern the process are beautifully simplistic. The implications are far-reaching. We're going to have a look at normal cell division that an animal uses to grow, repair wounds. This is known as mitosis, where every single diploid cell which divides produces identical diploid daughter cells, and they are identical in every respect. Once we understand this process, you will also understand the importance of the difference between that and meiosis. So we're going to start by looking at mitosis, and we're going to compare and contrast that with meiosis, which an animal uses to produce cells for sexual reproduction. And this is when our diploid animal produces haploid cells that combine during fertilization with another haploid cell and reproduces a diploid animal. This is a very necessary part of sexual reproduction, and it is what ensures that all of our offspring in any given clutch are actually unique, and they will all contain different combinations of the possible genetic mutations that are present in both the parents. We're also going to use our knowledge of that process to look at a fairly controversial topic: temperature sex determination. We're going to have a look at that and try to decide if temperature sex determination is a real thing in our ball pythons. And we're going to use a classic example of genotypic sex determination, which is actually the banana sex link, and we're going to look at how that works at the cellular level. So if you're ready for this, let's get after it. So we need a couple more concepts here. Ploidy is the number of complete sets of chromosomes in a cell, and our ball pythons are diploid, which means they have two sets of chromosomes, one from one parent and one from the other, and they exist in homologous chromosome pairs. There are actually 17 homologous pairs and one pair of chromosomes which is XY, which determines the sex of the animals. Under normal conditions, the haploid number, which is one set of chromosomes produced by a process known as meiosis, which is the process that makes eggs and sperm, there are 18 chromosomes in a haploid cell, which is 18 chromosomes in an egg or 18 chromosomes in sperm. For diploid organisms, one set of chromosomes is what we get in the monoploid cell, and that's the same number of chromosomes as we get in a haploid cell. The difference with haploid cells, as we shall see in a moment, is that the process of meiosis means that they are not duplicate sets of any one parent. They actually have exchanged some genetic code between the homologous pairs, and haploid cells are unique.
It's a process exclusive to sexual reproduction, and the haploid cells fuse with another set of 18 chromosomes from the second parent during fertilization. So the embryo produced is again a diploid animal, and in our ball pythons it will have 36 chromosomes, two sets of 18 chromosomes, one set from each of the fused haploid cells produced by each parent when they make their eggs and sperm.
Alright guys, let's deal with cell division and how cells replicate. This is not complicated, at least not in this presentation, but it is extremely important. So, we know that our total DNA strand, half of it from dad and half of it from mum,
is divided into 17 homologous pairs of chromosomes plus an additional pair, the XY chromosome, which are not identical. So 18 pairs of chromosomes in total in this diploid cell.
And if we look at each individual chromosome, the process that I'm going to describe now is replicated in every single one of those 18 pairs of chromosomes within the cell. So let's assume that we're dealing with a snake that has one genetic mutation. Let's say it's Enchi. And Enchi was originally from the dad, and Enchi resides at this location here. Now, these chromosomes are still homologous. This side of the chromosome contains the same gene, but without the genetic mutation that causes Enchi. So this is the heterozygous form of the Enchi allelomorph or genetic mutation. Doesn't really matter within the cell. Once we've recombined the genetic material from mum and dad into a diploid organism, it doesn't really matter whether the genetic mutation is associated with mum or associated with dad. So when we want to divide cells, when the animal needs to grow, when the animal needs to repair a wound, it needs to replicate cells. And each of these homologous chromosomes divides. So each side of the diploid has exactly replicated itself. We've got an exact copy of this side, which is carrying the genetic mutation or allelomorph for Enchi, and we've got an exact copy on this side of the diploid of the chromosome that is not carrying the genetic mutation. So, in the first phase of mitosis, we sort all the chromosomes into homologous pairs, and each of the homologous pairs duplicates itself, and these are known as sister chromatids. Science recognizes this as still a single chromosome and this as still a single chromosome, but as you can see, they're actually identical copies of the same chromosome on each side of the diploid. So we've created an exact copy of each of these homologous chromosomes. When the cell divides, these line up, and when they are pulled apart as the cell divides, you can see that we now have two identical copies of the original chromosome. Each of the 18 chromosomes does exactly the same thing. We have produced two daughter cells which are identical to the original. So we go from this to this to this. This original diploid cell with its 18 chromosomes replicates every single one of those chromosomes and ends up with identical copies of each homologous chromosome in 2 identical daughter cells. So we've gone from 1 copy to 2 copies. The cell has replicated itself. Alright guys, you've seen Mr. DNA presentations before where I've talked about how we split the genetic material in half from both parents and then recombine it in the offspring. How does that happen? Let's get down and dirty, let's take it to the cellular level, and let's have a look in depth at some of the processes that go on when we breed our ball pythons. Okay, what happens when we want to make gametes, when the female wants to make her egg cells and the male wants to make his sperm cells? It wouldn't be good if we created exact copies of the parent. That would mean that there is absolutely no variation between parent and offspring. So there's no possibility of the species adapting, for instance, some variations being better adapted to a new environment and therefore preferentially passing on their genetic composition. So the process where we produce gametes is actually a little bit different, but only in detail. So here we go. We're looking at an individual chromosome, and this chromosome, as before, happens to carry on one side of the homologous chromosome is carrying the genetic mutation for Enchi. The other side of the homologous chromosome at a different locus is carrying a different genetic mutation. It is carrying the pastel gene. Both of these are heterozygous for Enchi, single copy, and heterozygous for pastel, single copy. So this is actually a pastel Enchi. We don't know exactly where this genetic mutation resides or on what chromosome. So this is schematic. I've assumed it is on one chromosome at a different locus on each side of the homologous pair. So the process of cell division is exactly the same to begin with. Each of these chromosomes duplicates itself. So the chromosome on this side has duplicated itself exactly. They are identical in every respect, and you can see that we have replicated the genetic mutation, which is Enchi. On the other side of the chromosome, we have again duplicated that chromosome, and we've duplicated the genetic mutation, which is pastel. And normally, if this was mitosis, we would split these up and we would realign them like that and have identical copies. But we don't actually want to do that in sexual reproduction because all the offspring will be identical to the parents. So a slightly different process occurs. These sister chromatids actually line up. They all bundle together, they all line up, and remember that these are identical chromosomes in every respect. So what that means is that this genetic mutation is actually free to move to any free slot on any of these 4 chromosomes. So this one is free to move between these 4. This genetic mutation here for pastel is free to move to any other free slot because they're sitting right alongside themselves. It's easy to juxtapose. So what we're doing is actually shuffling small pieces of DNA across these 4 chromosomes. When we split them up, we're going to split them up into haploids. We're going to create 4 haploid cells rather than 2 diploid daughter cells. The diploid daughter cells would be identical to each other, and we've already said we don't want to do that in sexual reproduction. We want to have little pieces of each of the parents incorporated into the offspring with the possibility of some variation. So we split these now into 4 separate haploid cells, which are sperm from a male and egg from a female, and we can now see that each of these haploids is unique. This one carries no genetic mutations. This one carries a single copy of the pastel allelomorph or genetic mutation. This haploid carries both an enchi and a pastel, and this haploid carries a single copy of enchi. So you can see that each of these sperm cells or each of these egg cells, depending upon whether it comes from a male or a female, is unique. That is where all the genetic permutations that you will see on genetic calculators comes from. It is part of the meiosis splitting process that mixes and matches little pieces of DNA across the chromosomes, and each of these haploid cells or gametes is unique. They are not identical to the parent cells. That's why in any given clutch the genotype of each of the offspring is going to be different. This is from one parent. Imagine the genetic mutations that you would get from the other parent, which is also producing haploid cells. When you recombine them, the permutations are statistically derived based upon what combinations are in each of the parents, so it is predictable, but they are all going to be unique. They will all have little pieces from both parents. Not just the colour and pattern mutations, but other genetic mutations as well, which could mean slight differences in the ability of a snake to survive in nature. This is essential for adaptation of a species during evolution. It's how a species evolves, it's how a species gets better. It's also how we produce all our different genetic mutations. Phew, if you got through that, mitosis versus meiosis, you're going to find the next bit extremely interesting. As I said, nowhere near as complicated as the scientific terms would have you believe. The principles of mitosis and meiosis are actually very simple. Let's have a look at how temperature might affect the development of our embryos during the time that the eggs are under incubation. Now you'll notice, guys, when I candle these eggs here, you will notice the blood vein network in here, which indicates that this guy has already undergone quite a lot of cell division. There is an embryo in here.
Okay, temperature sex determination as opposed to genotypic or genetic sex determination is when environmental conditions, particularly temperature, control the sex of the offspring in any particular clutch. Temperature sex determination is observed in some reptiles and some fish, but it's actually quite uncommon among vertebrates. Chromosomal sex determination or genetic sex determination is much more common among vertebrates. Now, the important takeaway here is that there is a thermosensitive period. There is a particular period during egg development when temperature is particularly important in determining the sex of the offspring. And this is usually the middle third of incubation. And I've highlighted in yellow at the bottom that development of the embryo includes the time it spends in the oviduct before the eggs are laid. And in this case, for our ball pythons, that is 50 days plus inside the female after ovulation, but before the eggs are actually laid. There are two systems of genetic or chromosomal sex determination. One is the familiar XY system, where the male is the heterogametic sex, he is the XY animal, and the female is the XX animal, and it's the male that determines sex of the offspring. The other system, which is common in some reptiles and birds, is the ZW system, and here it's the female that is the heterogametic sex. She has the Z and W chromosomes. The male only supplies ZZ. So fertilization by the male does not determine the sex of the offspring in the ZW system. In many reptiles which display temperature sensitive sex determination, the sex chromosomes are indeterminate. Many species with ZW also have temperature sensitive sex determination, but it's quite rare for XY chromosomes to have temperature sex determination. Our ball pythons are, of course, XY animals, just like humans. This, of course, means that it is much more likely that the chromosomes are controlling hormone production during the development of the embryo rather than temperatures. In genotypic sex determination, it's these chromosomes which control the hormones, which control the production of the enzyme aromatase. Aromatase converts androgens or male hormones to estrogens or female hormones. So the more aromatase you have, the more likely it is you're going to get females. Temperature, of course, influences the production of aromatase. And so it's the enzyme aromatase that actually is temperature sensitive, and that determines the sex in our TSD animals. Another common misconception about temperature sex determination is that there is only one kind, and a common misperception is that cooler temperatures equal more males and hotter temperatures equal more females. That is not actually true. It's true of turtles, but it is not true of lizards. Lizards are actually the other way round. And in crocodilians, we see temperatures at the two extremes, cool temperatures and warm temperatures producing females, whereas medial temperatures produce more males. In all three of these diagrams, we only get a mix of sexes in a very narrow temperature range, very close to the threshold. Temperatures above and temperatures below produce almost exclusively all males or all females. So I've summarized that in this slide so you can pause it and read all the details. Cooler temperatures producing males occurs in turtles. Hotter temperatures producing males occurs in lizards. I think lizards are generally considered to be more closely related to snakes than turtles are. So would we actually expect more males to be produced at cooler temperatures or more males to be produced at hotter temperatures? The point here is that there is actually several different systems, none of which appear to be associated with our ball pythons. So, based on what we've learned so far, cell division after fertilization is by mitosis, and all the diploid daughter cells are identical to the fertilized diploid parent cell, which of course includes the XY chromosome. Fertilization is the time that both sex and the genotype of our ball pythons is determined. So when the female ovulates and eggs meet sperm, both the sex and the morph of each individual snake is already determined. Now, that's not to say that there might not be some temperature influence during the breeding season. Male and female sperm, for instance, may have different degrees of durability. We know, for instance, in humans that male sperm swims faster but doesn't last as long, whereas female sperm has a lot more staying power. So it could be that during meiosis, when a male makes his sperm, or after mating when the female stores the sperm, that temperature effects may be actually selectively killing either male or female sperm. We know, for instance, that females will lay slugs if the male or the female has been subjected to high temperatures during the breeding season when she has successfully paired multiple times. But this effect, of course, has nothing to do with incubation temperatures. It has everything to do with the way that our snakes are kept during the breeding season, when females are mating with males and when males are producing their sperm. There could also be an effect of timing in relation to ovulation. The longer that a female has to store sperm, we may actually be getting selectivity in our sex ratios because of the time between successful mating and ovulation. Maybe female sperm lasts that bit longer than male sperm when it's stored. This, of course, is going to vary tremendously from breeder to breeder, depending upon his hotspots, his substrate, his tub configuration, and it's also going to vary between pairings of individual snakes. We cannot guarantee the length of time it takes between the last pairing and the time the female ovulates. All of these factors are much more likely to produce a variation in sex ratios than any difference in incubation temperatures. Without rigorous control data and elimination of all these other possible variables that could affect sex ratio, a comparison of incubation temperatures and sex ratios is absolutely meaningless.
And there is a beating heart. If there are blood veins, there must be something pumping the blood. So this guy has already got a heart, which means that cell division is advanced enough to the stage where we're getting specialist organ cells rather than just straight cell division. We are actually getting specialized cells. They're already divided into functions, and that happens inside the female after ovulation. These eggs have been inside her for 50 days. They're already quite well advanced. Remember, these eggs have already got a beating heart. They've already got specialized cells divided into organs. They're already 50 days old, and they're not even in the incubator yet. So how much influence is our incubator temperatures going to have on the development of these eggs? Okay, now we understand a little bit more about temperature sex determination and some of the processes that might be involved. I think it becomes fairly clear that temperature of our incubators in all cases makes absolutely no difference to the outcome. Now, that isn't to say that there isn't a temperature influence earlier on in the process. I have described meiosis, and that can happen weeks or months before the eggs are even laid, and it's during this time period that temperature may actually have an influence over the outcome. You cannot change a Y chromosome to an X chromosome simply by incubating at a higher temperature. What may be happening is that when sperm is generated and when sperm is stored by our females, temperature has a preferential effect on which sperm survives the longest, and therefore preferentially affects which sex of sperm may encounter the eggs at the time they are fertilized. It could be there are more X chromosome sperms or more Y chromosome sperms available to fertilize the eggs at the time that ovulation occurs. Once that is done, the sex is predetermined. We're going to have a look at the banana sex-linked gene. The banana gene just happens to reside on that chromosome out of the 18 pairs of chromosomes in our ball pythons. It resides on the sex chromosome. It resides on either the X or the Y. As I've already described, in the process of meiosis, we cannot change a Y chromosome to an X chromosome with temperature. So let's have a look at why the banana gene is sex linked and why we get male bananas which are male makers and male bananas which are female makers. How does that work? I'm going to talk about the banana sex link, and the banana sex link everybody has heard about males being male makers or female makers, and I'm going to explain how that works on the cellular level. It's worth taking some time over this because the banana sex link is the very definition of genotypical sex determination. Now, we know in our ball pythons that we have diploid animals, and there are 18 pairs of chromosomes in each cell. 17 pairs of those chromosomes are homologous or the same, they're mirrored on each side of the diploid. 2 of those chromosomes are not homologous, they are different, and they are the XY chromosomes. And in ball pythons and in humans, the male animal is the XY chromosome animal and the female is the XX animal. When we make our gametes through meiosis, the female can only give an X chromosome. The male gives 50% of his offspring a Y chromosome and they will end up being males, and 50% of his offspring get the X chromosome, and when that combines with the X chromosome from the female, they are females. Okay, so let's have a look at our male banana. And the banana gene actually occupies a locus on the sex chromosome, either the X or the Y, and this is important because we know that the X and Y chromosomes are not homologous chromosomes, and the banana gene happens to be associated with the sex chromosome. So our male is an XY animal, and I'm going to put the banana gene on his Y chromosome. This would make him a male maker. Let me show you how that works. Okay, so in meiosis we know that each of these pairs of chromosomes first of all replicates itself. So the male will make an exact copy of his Y chromosome with the banana gene attached to it, and this is the daughter chromatid. And his X chromosome duplicates itself, and the X chromosome is not carrying the banana gene. Now, in normal meiosis, all of these 4 chromosomes line up, and in any other chromosome, the locus for that particular gene lines up. So the genetic mutation is free to move across any of those 4 slots. But the X and the Y chromosome are not the same, so it doesn't work quite the same way as for the other 17 pairs. The X and the Y chromosome are a little bit different. So when they line up, the slot for banana is not juxtaposed with the slot for banana between the X and the Y chromosome. So if the banana gene is attached to the Y chromosome at this slot, it is, when the genes line up, it is very difficult for this genetic mutation to make the jump across to the X chromosome. So difficult, in fact, that it very rarely happens. If the banana gene is attached to the Y chromosome, only if there is a crossover event in special circumstances, about 20% of the time, if you are lucky, this gene will make the jump across to the X chromosome. But that does not happen most of the time. So when we create the 4 haploid cells, when these split up, the 2 female haploid cells or gametes do not have the banana gene. They could not get a copy of the banana gene during meiosis because the locus does not line up exactly, and it's very difficult for the gene to make the jump. Out of the 4 haploid cells, 2 of which are male, both of these have the banana gene. So banana is usually, in almost all circumstances, passed on with the Y chromosome in a male maker. All of his banana offsprings will be males, and all of his male offspring will be bananas. That is the very definition of genotypic sex determination. Let's have a look at a female maker. So in this particular case, this male banana is actually the son of a female banana, and the banana gene is attached to the X chromosome. So here we go again. Meiosis. Each of these pairs of chromosomes duplicates itself, to produce the sister chromatids. These line up, and again, when we line them up, because these chromosomes are not the same, the locus for the banana gene does not line up with the locus for the banana gene on the Y chromosome side. So again, when we create our 4 haploid cells, the 2 Y chromosomes from the female maker are not carrying the banana gene. The 2 X chromosome haploids are carrying the banana gene, and during the process of meiosis it was almost impossible for them to make a jump across the locus because the chromosomes do not line up exactly during meiosis. So for a female maker, every single female is banana and every banana is female. Again, the very definition of genotypical sex determination. Let's just do that for a female banana. The female banana is an XX animal, and she carries 1 copy of the banana gene on one side of that sex chromosome pair, and in this case it's XX. So what happens when she makes her egg cells? Each one of the pair of sex chromosomes duplicates itself. The one on this side, on the sister chromatid, is carrying the banana gene. And the one on this side, and I've run out of pink, so we're going to have to use red. But the sister chromatids on the other side of this XX pair does not carry the banana gene. When they line up, these do actually line up exactly, and the banana gene is free to move across the locus to any one of these 4 chromosomes, but it doesn't matter because they're all X chromosomes. So when they're pulled apart in a female, we end up with 2 haploids, X chromosome with no banana gene. 2 haploids with the banana gene, both X chromosomes. Each of these haploids can receive an X or a Y chromosome from the male. Remember, it's the male that determines the sex. So 50% of these will get a Y chromosome and they'll be males, and 50% of them will get an X chromosome, so they'll be females. So you will have exactly 50-50 males to females, exactly 50-50 bananas to non-bananas, and exactly half of those bananas will be males and half of them will be females. And that's why, because of the nature of meiosis, exactly the same process in the female means that females always give 50-50 sex ratios in their banana offspring. Their offspring will always have the banana gene associated with the X chromosome, so any males from her will be female makers. Okay guys, I hope you're still with us here and that you were able to follow that. As I say, it appears on the surface to be a very complex subject, but actually the scientific principles that govern it are very simple. In the next episode, the final part of this genetic series, we're going to use this knowledge of cell division and how meiosis works and look at some of the unfortunate side effects of these processes when they go wrong. We're going to look at parthenogenesis and we're going to look at genetic deformities. So stay tuned for that. Thanks for watching. Don't forget to share, like and subscribe. Links are all in the description below, guys. All the references for you to study away to your heart's content. We'll see you next time.